And now I'm happy to introduce Dr. Ryan Spitler. He did his postdoc at Stanford and he became the deputy director of the FIND program on precision health and Canary Center. And in that context, he worked very closely with Sam Gambier. So please welcome Ryan, who's going to introduce to you also my colleague, Dr. Chris Contag. Thank you.
Thanks so much, Brian. Yes, I'm Ryan Spitler, and yes, I am Deputy Director of the Precision Health Integrated Diagnostics Center, as well as the Canary Center for Cancer Early Detection. Many fond memories of Sam. Sam and I founded the Precision Health Integrated Diagnostics Center together in 2017, based on his long-term precision health vision, which he had been thinking of for many years before this. And starting initially with just the two of us, we were able to build the program, which has now launched over 50 projects to date and is spanning the entire Stanford campus. And we've been able to collaborate in such major initiatives as the Project Baseline Health Study, which, as many of you know, is one of the most, if not the most in-depth study of human health ever undertaken. I feel very fortunate to have had the privilege to be able to work closely with Sam during the last several years, and forever will be inspired by his passion and vision. So thank you, Sam, for having such a profound influence on all of us. And next, very excited to introduce Professor Christopher Contag. Dr. Contag joined Michigan State University in 2017 as the founding director of the Institute of Quantitative Health Science Engineering, IQ, and the inaugural chair of the Department of Biomedical Engineering. He is a professor of microbiology and molecular genetics and biomedical engineering. Dr. Contag received his BS in biology and his PhD in microbiology from the University of Minnesota. He was a postdoctoral fellow at Stanford University from 1990 to 1994 in microbiology, and then joined the faculty in pediatrics at Stanford in 1995. At Stanford, Dr. Contag served as the Associate Chief of the Division of Neonatal and Developmental Medicine, the Director of Stanford's Center for Innovation and In Vivo Imaging, Sci3, and Co-Director of both the Molecular Imaging Program at Stanford MIPS and the Child Health Research Institute. Dr. Contag is Professor Emeritus at Stanford University. He founded the companies Xenogen, Pixel Gear, BioEclipse, and Exoforce. So pleasure to have you today, Chris, and I'll turn it over to you.
Thanks, Ryan. Thanks for that kind introduction, and really nice to see you again, Ryan. It's been a few years. It's also nice to see all of my colleagues from Stanford. It's nice to hear your work, and it was really tremendous listening to Gary talk about how the vision that Sam had so many years ago is really being expanded and some of the goals he laid out in the very beginning fulfilled. So it's really wonderful to see all of that today. I want to thank Joe for inviting me to speak in this tribute. It's definitely bittersweet. I spent many years working with Sam and miss him dearly. And every day, I think he guides my science and helps me mentor students. And in my seminars, his influence is always felt and his spirit is always there. And I guess many of us on the call today can say that because his impact has reached across disciplines and around the globe. And all of us really owe a tremendous amount to Sam and his vision. And I know I do in particular. And I really appreciate all of you coming together for this tribute. So what I'd like to talk about today, as I mentioned on the very first slide, we think of the Institute as first ever to first in humans. And I'd like to focus on the first ever end of that continuum. We've developed a number of, the community in general has developed a number of ways of engineering biological systems. And I'd like to offer today an alternative kind of based on a what if question. What if we could recapitulate eukaryotic evolution and engineer organelles from otherwise free-living bacteria? What's really great about this is when I was at Stanford, Ryan and I were working on this with Brian Rutt and continuing to work on this, but this is the first time I've really spoken about it in a large forum, and it's because a number of the papers have finally come out after years of trying to get to this point. So when I asked students this question, what if, we hear things like, would it take us 1.5 billion years to develop, just like it did for evolution? And would we need another 2.7 billion years to refine it, again, like evolution? And my answer is I hope not. I'm hoping we can build on the 4 billion years of prokaryotic evolution to find the tools that allow us to recapitulate what evolution took billions of years to do. And then we ask, what if? What if we could use the synthetic or engineered organelles as biologically encoded remote control modules to direct biology? This would offer transient control that would preserve the host genome and allow us to redirect a cell and reprogram it, and then eliminate the remote control module when it was no longer needed. So what would you do if you had a remote control module that could direct biology? And this is what I'd like to spend a little bit of time talking about today. You think about the barriers we need to overcome to generate synthetic endosymbionts and engineer them for cellular control. The first thing we'd need to do is get the bacterium into the cell. And what I'll show you today is we can introduce this via phagocytosis, but it can also be introduced with cationic lipids or with biological assistance from the lessons learned from other intracellular bacteria. But then once the bacterium is inside the cell, it needs to break out of the phagolysosome and into the cell cytoplasm. And then we need to flip another switch to turn on a mammalian transcription factor to control the cell program. And we could do this either with metabolic or magnetothermal induction. Then we'd need to program that protein to be exported out of the bacterium and into the cytoplasm of the cell, and then engineer that protein to also get into the nucleus of the host cell, such that it could affect the transcription patterns of this cell and change the cellular phenotype or function to a desired function that would serve our purposes. The other part of this that others have worked on, and I'm not going to talk about today, is to engineer codependence for symbiosis so that the bacterium would persist for periods of time, much like a mitochondria would. Others, like I said, have worked on this as an example from Peter Schultz's lab using yeast and E. coli and forcing a endosymbiotic relationship by tying their metabolism together. And tools like this could be used to drive endosymbiogenesis in mammalian cells as well. So the first step, as I said, is to engineer endosymbionts and get them inside host cells. And we started with the bacterium Bacillus subtilis, which is a gram-positive rod, and it's engineered to express the stereolysin O, which is an enzyme that allows listeria to get out of the endosome of a macrophage. This was kindly provided by Dan Portnoy at UC Berkeley. What I'm showing here is if we stain for membranes in green, the nucleus in blue, and the endosome in red, you can see the engineered endosymbionts that we call EES in the cytoplasm of the cell. Not only are they inside the cell, but you can see them over time here at 1 hour in the cytoplasm and then replicating over 4 hours. So it's possible to get the bacteria into the cell and to have that cell survive as well as the bacteria replicate. These studies have been summarized. I'm just going to have with a short period of time, just a few minutes to touch on a few of the results, but this is available on bioarchives if you'd like to look at a paper by Cody Madsen and others in the lab who have advanced the use of these tools. The macrophage survives even at very high MOIs of 50 to 1 bacteria to mammalian cell until you get to these higher MOIs in longer periods of time. So with the right number of bacteria designed correctly, you can get them inside the cell and have them persist. We can also express from the bacterium beta-galactosidase under a mannose-regulated promoter, so we can flip it on and off with adding a sugar that's not metabolized by mammalian cells. And we can show that the beta-galactosidase with a nuclear localization signal targets the nucleus. And here we're comparing mannose induction, but no nuclear localization signal, to mannose induction with a nuclear localization signal, showing an increase in the number of the amount of protein per nuclei. And here it's quantified to make it a little easier to interpret these images. So by flipping the gene on and having it exported through a TAT secretion system out of the bacterium into the cytoplasm, and then using a nuclear localization signal, we can get this reporter gene to target the nucleus of the host cell. To see this more clearly, we did stain this with an antibody, and this explains the red color. We can also look at the distribution of these bioluminescent bacillus cells, or these engineered endosymbionts, and see that they go to the liver, suggesting that we'd be able to target the macrophages or the Kupffer cells in the liver with these endosymbionts and then redirect them or reprogram them. There are also studies showing that these bacteria go to tumors, and I'll show you that in just a moment. So with this background information and these studies, we engineered these bacterial operons comprised of mammalian transcription factors. And when you see that phrase written, you wonder, it seems insane that you would express mammalian transcription factors from a bacterial operon. But the point was to try to reprogram macrophages from M0 to M1 or M2. So again, using the mannose promoter and in an operon that expresses two transcription factors that drive an M1 phenotype, or alternatively, two transcription factors that drive an M2 phenotype, these bacteria could be introduced in macrophages and either drive a pro-inflammatory response if you wanted to treat a cancer, or an anti-inflammatory response if you wanted to treat autoimmune disease, as examples of ways of reprogramming cells for the benefit of human health. So in this example, you can see that we changed the surface expression of macrophages by using a SK plus the mannose induction, and this is compared to the M1 control. Like I said, I'm just showing you examples of how no treatment and then induction of the transcription factors leads to an M1 expression, and that diminishes over time. Alternatively, if we look at the cytokine profiles, we can see that even though Bacillus subtilis with LLO alone induces some cytokines, when you express the KG transcription factors, which drives an M2 phenotype, you see an increase in anti-inflammatory cytokines. And again, you see a difference here with mannose control. The regulation isn't as tight as we'd like, so you do see some induction, even if you're not, or some expression of these transcription factors, even if they're not being induced with mannose. So we like the sugar control, but what we observed in these studies is that mannose itself drives a pro-inflammatory phenotype. And so we wanted to work on another approach towards regulating these transcription factors. And for that, we've continued our work on magnetotactic bacteria. Magnetotactic bacteria biomineralize iron into iron oxide and forms these little nanomagnets. And those can be imaged by magnetic particle imaging. But in magnetic particle imaging, the signal is dependent upon the configuration of the iron oxide particles. And so using a mutant from the Cellular lab, MSR1, where the biomineralized iron particles group at the end of the bacterial cell, we get a much brighter signal in MPI. So Sam was really the consummate chess player, and he would look at a situation and he would calculate where the field might be in 10, 20 years. And so back in 2009, he published a paper along with A.C. Mateen at Stanford, showing that you could image these magnetotactic bacteria with MRI, and this shows that study. A few years later, in 2018, working with Ryan and Brian Rutt and Caleb Bell and Kim Brewer, we showed that we could detect these cells loaded with the magnetotactic bacteria. So mammalian cells loaded with the magnetotactic bacteria, looking at 100 cells or 1,000 cells injected into the brain of a mouse. So this demonstrated that if we could get the operons into these cells, we could then watch where these bacteria were by magnetic particle imaging. And so we started looking at MSR1 and the signals where this bacteria would localize over time. And we labeled the bacteria with both luciferase expression and with looking at the magnetic particles in these cells using MPI. The two modalities show different things, in that when you have distributed signal, the signal's not concentrated in any one place brightly enough to have a signal above background. But by the time the signal concentrates in the liver, it's too deep and the liver's too dark to see with a luminescent signal, and the MPI shows you that it's in the liver, again suggesting that we could target the liver for control. The iron oxide offers another opportunity as well, and that opportunity is to control the expression of genetic elements using alternating magnetic fields. So the idea would be that we could put a thermal switch, and these are thermal switches that were developed by Mikhail Shapiro at Caltech, in front of essentially a light bulb, a biological light bulb. And by activating the gene, by heating the iron up with alternating magnetic field, we could turn the genes on and demonstrate that we could activate a luminescent signal. And if this was successful, then we could replace this operon, this 5-gene luciferase operon, with multiple genes for transcriptional regulation in mammalian cells. So Emily Greason in the lab, and again, her paper's available at bioarchives, showed that she could activate the expression from the magnetotactic bacteria with alternating magnetic fields, showing that the Lux gene can be turned on with different exposures to different temperatures for different lengths of time. And that we could also coat Bacillus subtilis with iron oxide and flip the genes on with these thermal switches. So what that suggested to us is that we could create a variety of mammalian transcription factor operons that could control a variety of reprogramming processes. Here's the collection we started to work on, including operons that would allow us to reprogram cells into iPSCs and then drive them into cardiomyocytes, hepatocytes, pancreatic cells, or skeletal cells and neurons. And if we could do this with the magnetotactic bacteria and the alternating magnetic fields, we should be able to drive tissue engineering from outside the body using these combination of tools. So with that, I'd just like to summarize and say that magnetothermal control would allow external regulation and guidance of regenerative processes. We can watch these with MPI, MRI, and CT, as well as ELI in our mouse models. Bacteria therapy is currently the standard of care for bladder cancer, so we're already using live bacteria as a therapeutic, and it is emerging as a means of delivering complex programs to treat complex diseases. So the idea of using these as endosymbionts as opposed to exogenous bacteria outside the cell isn't outrageous. And if we could generate complex transient therapies that would restore tissue homeostasis or regenerate tissues and guide tissue responses, we'd be able to rebuild damaged tissues and regenerate lost function. So there are a variety of regulatory mechanisms in prokaryotes that will enable fine control in response to changing tissue environments. So it could be that they respond to an environment where these programs need to be activated, and that would enable dynamic therapeutic approaches. With that, I'd like to thank all of you for your time and listening for today, and all the people in my group that have made this possible. The ones shown in red are the ones contributed to the study today. I thought it was cute to have a little Harry Potter-esque picture of the group. We've got a great group of students and postdocs. A lot of the work you saw today was from Cody and Emily. They're both graduating this summer, and Ashley, who's a postdoc. We've had great collaborators across a variety of different areas, from microbiology to imaging tools. So I want to thank everybody again, and I'm happy to take any questions.
Excellent. Well, thanks so much, Chris. That was a very interesting talk, and great to see a lot of follow-up and progress. I know a lot of those mechanisms took a lot of fine-tuning by us and others, so I'm glad to see it moving forward. I'll start us off with some questions, but feel free, everyone, to please use the Slido to add your questions as well. I know one of the things, a lot of the projects you'd go after, you'd like to try to go, really shoot for the stars. We could do the simple things as the controls, but let's really go for it. And I remember one of the things that you mentioned to me in the beginning was, can we, de-differentiate cells so that you could eventually, create organs internally if you're repairing tissues or such. Is there still some ongoing work in that area that you're considering beyond like the applications you mentioned?
Yeah, I think the real strength of this approach, given the inefficiency of bacteria being taken up by mammalian cells is to target something we can hit a small number of cells and have those cells then turn into a tissue or an organ. So I think controlling stem cell function in biology with engineered endosymbionts is the real sweet spot for this technology. I think it has applications also in treating tumors and just bacteria therapies. But the idea of controlling genetic programs with engineered endosymbionts, I think, is the real strength will be in stem cell biology and tissue regeneration.
Yeah, I know like some of the power of this is also the ability to regulate these in a controllable way. And as you mentioned, it can be leaky in some cases. I know in some previously we thought about, using things like Rago thermometers, especially.
Quite good at regulating gene expression. The challenges we have there are transferring them from a gram negative to a gram positive. The other challenges we have are using the alternating magnetic field to get the exact right temperature. So in the thermal cycle, it works beautifully. The regulation is pretty tight, predictable. But when you move to the variation with different numbers of iron oxide particles and where the energy is deposited, that's a challenge. The other challenge is that it takes some time to warm these promoters up to melt the repressor off sufficiently to get gene activation. And so we're trying to shorten that up as much as possible. And that's going to take a little bit of engineering. And that's where we're working with Mikhail Shapiro on some of these repressor models.
And one of the nice things also about this was you have the kind of the dual ability to turn genes on, but then you also can track, wherever the cells have this in them. I was curious. I mean, you can use MPI to track them as you showed. Do you have any changes in expression, given that that's also kind of a hysteresis-based mechanism? Or is it, to your point, you just said that it'd be too brief and you wouldn't have to worry about that at this stage?
Yeah, it's way too brief. I'd be happy if we'd see some gene activation in the MPI. The alternating magnetic field generator we're using for now a period of 60 minutes or so, and so that's just not practical, and that's not the energy, the duration of energy that you see in the MPI. So yeah, you're not going to see activation at those short duration.
Okay, great. Let's get some questions from the Slido as well. One question is, how did the particles produced by magnetactic bacteria compare to that of commercial particles or particles optimized for MPI in terms of PSF and FWHM?
Yeah, they're surprisingly close to the particles that we typically use. And so one reason that we could coat bacillus with these iron oxide particles that are commercially available and have them try to mimic what we see with the magnetotactic bacteria is that the structures are very similar. And the signals that we can generate are very similar as well. So surprisingly similar is the short answer.
Okay. Another question is, given that macrophages are thought to be more of a continuum from M1 to M2, depending in part of their milieu, rather than binary M1, M2, how does that enter into the idea of controlling their polarization? And can these technologies allow a readout to help measure their polarization in vivo?
That's a really great question. And I didn't spend too much time on the fact that it's continuum and not binary. We selected the 2 transcription factors for each of these cassettes to try to keep it relatively simple so that we could drive what looked like an M1 or what looked like an M2. But we've done the complete cytokine profiling and cell surface markers. And it's obvious that we haven't created a binary situation with M1 and M2, but rather activated some cytokines that we would expect and others that we were surprised to see. And so we do see a collection. I don't think that imaging tools are going to allow us to watch that in vivo. So it'll allow us to turn things on. We can say, yes, this operon is on. This operon drives a M1 phenotype, we could say that, but we don't know that that phenotype was achieved using imaging tools. So we recovered the macrophages and profile them with flow cytometry or cytokine profiling.
Excellent. Well, thanks so much, Chris. It was really great to see you again, and it was, always a pleasure to see all the exciting work that you're working on. So I hope to stay in touch and look forward to continued excellent things.
Thanks, Ryan. It's nice to see you, and thanks everybody for attending.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.